In the previous video, we talked about the basic chemistry related to biological system. Now we are going to look at the main macromolecules that makes up the body. There are four main macromolecules in the body. They are carbohydrates, proteins, lipids, and nucleic acids. We can think of macromolecules as polymers. Polymers are basically really long molecules that are consisted of repeating units that are called monomers. Okay, so you can think of it as a long train. Okay, the individual parts of the train are the monomers, and the entire train would be the polymer. And different macromolecules will have different names for their polymers form, and they are consist of different types of monomers. And we'll be looking at this in more details in the slides coming up. So this is a generic example. We have two monomers here, and all the monomers that we're going to talk about in the macromolecules, they're going to have some kind of OH group in it. Okay, so when two monomer comes together, then an H is going to be removed from one of them, and then the OH is going to be removed from the other one, and as a result, the two monomers are going to be connected together in a covalent bond, and you are going to produce water as a result of that. Because you are removing a water molecule from the monomers to join them together, this type of reaction is called a dehydration synthesis. You're creating something, creating a bond by dehydrating the molecules. It's also known as a condensation reaction, because the water molecules is being removed. So multiple monomers can join together to form a polymer. You can also break polymers down. So if you ingest carbohydrates, right, your digestive system is going to break the polymers back into monomers, so you can use it as energy, and it will be the opposite of the dehydration synthesis. Okay, to form the bond, you need to remove a water molecule. So to break a bond, you would have to do the opposite. You would have to add a water molecule. Okay, so if you add a water molecule to a polymer, then the H will go to one of them, and then the OH will go to the other, and it's going to split it apart. Okay, because you're splitting a bond with water, we call that hydrolysis. Hydro means water, lysis is to break. So essentially, just to summarize, polymer to monomer, that is going to be hydrolysis. And if you want to take monomers and make polymers again, that's going to be dehydration synthesis. Okay, and this could keep on going and going as long as you are adding water over here, and then this one you are removing water. Okay, so let's start by talking about carbohydrates. Carbohydrates is an organic molecule. In fact, all the macromolecules are considered as organic molecules because they contain carbon. So for carbohydrates, they always have carbon, hydrogen, and oxygen in it. Okay, and in fact, they are called carbohydrates because for every carbon in it, they would have the same ratio of hydrates in it. Right? Hydrate is basically water molecule. So this is a generic formula for carbohydrates, where N is any whole number. So if N is 6, then we would have C6, and then H2O 6, and we can put the 6 into the bracket. So we have C6H, 6 times 2 gives you 12, and then 6 times 1 gives you 6. And this, of course, is the formula we have been writing all lecture long. That is glucose. Okay. So if N is 12, then we will have C12H24 O12. Right. So all the carbohydrates tend to follow this general formula. Of course, there are exceptions, but typically speaking, they all follow this. Carbohydrates are important for providing quick short-term cellular energy. take glucose, for example, we can burn that relatively quickly and convert it into usable form of energy that can power our cell. We can use that energy to, say, move the muscle. Okay, we can use that energy to do transport work within the cell, and so that's one major function of carbohydrates. You can also find carbohydrates in various structural components in plants. For example, plants contain cellulose that gives them the fibers to support them. And in seashell, for example, there are some specialized form of carbohydrates. You might have heard of people take a glucosamine for their bones. So that glucosamine is that specialized carbohydrates that are derived from these seashells. So glucose is the preferred form of energy in the body, the C6H12O6 that we have been talking all along. We could take that and we can combine it with oxygen to produce carbon dioxide, water, and this process releases a tremendous amount of energy which we will be able to capture and convert it into a usable form called ATP. Okay, so we'll talk more about this ATP business in lecture 4 and 5. For now, just know that we can use glucose as fuel in the process called cellular respiration. And glucose is an example of carbohydrates. So where can we get carbohydrates in our diet? Well, there are many different ways you can get carbohydrates. We can get it in the form of starch. Right? There are starch in rice, corn, wheat, potato. So anytime you eat, say, pasta, eat bread, there is going to be intake of starch, right? Potatoes have a lot of starch in them. There are something called fructose. That's carbohydrates that you can find in fruits and in corn syrup. Lactose, you will be able to find that in milk. And then there are sucrose, which is kind of like your kitchen sugar, your table sugar, and you find a lot of sucrose in things like candy bars. Let's take a look at the chemical structure of carbohydrates a little bit closer. The monomers for carbohydrates, the building blocks for carbohydrates, are listed here. There's glucose, there's fructose, and then there are something called galactose. Okay, so chemically speaking, they are all C6H12O6, but then the arrangement of the atoms, they are a little bit different. Okay, so we call these isomers. Okay, not terribly important that you know this term, but for those of you who are interested, isomers are basically molecules that have the same formula, so they are all C6H12O6, but the configuration is different. So it's like having a Lego set, right? You can build a car with it, you can build a plane with it. They are made up of the same components, but they look different, right? So we call them isomers. Okay? So these single sugars, these glucose, fructose, and galactose, they are the building blocks for larger carbohydrates, and they are single sugar, so we call them monosaccharides. Saccharides means sugar, mono means single. So we can take two monosaccharides, okay, and we can combine them together. So let's say we take glucose. Right? Glucose is one of the monomers, and we take fructose, and you can see the glucose here has the OH and the fructose here has the OH as well. And if you want to connect them chemically, you will have to do dehydration synthesis, right? Also known as condensation reaction. In the reaction, you will be taking the hydrogen from the glucose and you will be taking the OH from the fructose. And when you remove these things together, they will form water. And after you remove the water, the glucose and the fructose will be linked together. So we will now call that sucrose. Okay, so this sucrose is a double sugar, so we call it a disaccharide. Okay, so if you take two monosaccharides and you link them together chemically through dehydration synthesis, you're going to end up having a disaccharide. And sucrose is what you will find in your kitchen sugar, right? And in candies. Here are some more examples of disaccharides, double sugar, right? And you can take a glucose molecule, combine it with another glucose, and that's going to give you maltose. You can find maltose in some syrup. You can have glucose plus galactose. Together, they will make lactose. So you'll find that in dairy products. And glucose plus fructose, we talked about already. That will give you sucrose. That is basically your regular sugar cubes. So all these are going to be dehydration synthesis, removing a water molecule to create the disaccharide. So you probably heard of people who have lactose intolerant. Okay, so when you take lactose, and again, the lactose is basically a glucose molecule connected to the galactose. And when you ingest this, your body wants to break the bond, right, by using hydrolysis to add the water and to break it down. But some people cannot do that. Okay, and so instead of breaking down the lactose and harnessing the energy that way, people who are lactose intolerant, they won't be able to do that, and the bacteria in their intestinal tracts will start to feed on the lactose, and that could result in gas productions, bloating, and diarrhea. So people can take these things called lactate, which basically is the lactase enzyme, a chemical that you need to break down this lactose. So normally you can make that in your body, and if you can't, then you can take the supplement before you go have some dairy products, and that will help you break down the lactose. So we talk about monosaccharide, single sugar, disaccharide, double sugar. Now let's talk about polysaccharides. So polysaccharides is the polymer of carbohydrates. Okay, these are really long chains that are either straight or branched. So straight means many glucose linked together like this. So there's one glucose, two glucose, so on and so forth, and it forms like a long chain. And sometimes it's branched. That means in addition to the linear chain of glucose, you might have branching points that are coming off like that, and it creates what we call complex carbohydrates, right? And so these often have hundreds and thousands of sugar monomers. They don't always have to be glucose. It could be a combination with other types of monomers. And common polysaccharides include the following. There are cellulose, glycogen, and starch. And we're going to explain what the function of these ones are on the slides that are coming up. All of these, the cellulose, the glucose, the cellulose, the glycogen, and the starch are made up of glucose monomers, and each one of them has different chemical properties, and that's because the glucose in them are connected differently. Let's start with cellulose. Cellulose is found in plant cell walls. The cell walls gives plant support and rigidity. It helps the plant kind of prop up, and a lot of these cellulose are actually undigestible in our digestive tract. So when we eat our veggies, we are ingesting all these cellulose, but we're not going to be able to break them down. And having dietary fiber is actually quite important to maintain good health, as I'm sure your doctor would tell you the same thing. Cellulose is a type of carbohydrates, and it makes up the fibers in the plant, like I said, and it is the most abundant carbohydrates on the planet, because there are a lot of plants on this planet, and so all these plants are going to contain a huge amount of cellulose. Even though humans cannot digest the fibers, a lot of the herbivores, plant-eating organisms, they can break down the cellulose. If you look at the guts of a cow, there are these specialized bacteria that they have in their digestive tracts that will help them digest the cellulose. If you ever look at a cow, they never really stop chewing, okay, and that's because the cellulose is really difficult to digest, and so they have to kind of chew them repeatedly in order to digest them properly. So next we have starch. Okay, starch is the storage form of energy in plants. The plants would use sunlight to make food, right? They do photosynthesis, and they convert water and carbon dioxide into sugar, into glucose, and that glucose will be connected together to form starch. Okay, so starch is branched polysaccharides. You can see here, there's the main branch, and then there are these side chains that are coming off from it, and the starch, we can actually consume them when we eat a potato. We are actually eating the storage form of energy in the plant, and we can chop up the individual glucose in the starch, and we use it for our own energy production. When we consume glucose, and if we have an excess of them, then we will convert it into our own storage form. And the storage form of energy in animals and humans are what we call glycogen. Okay, so we store glycogen in our muscle, and we also store glycogen in our liver. And liver is on the top right quadrant of your abdominal cavity, and you can see here the glycogen is even more extensively branched than the starch. Okay, and when you skip a meal, for example, and your blood sugar is running a little bit low, then your liver can take these glycogen and chop up a few glucose here and there, and you can release them into your bloodstream, and you'll be able to maintain the blood sugar level until you eat something. So to summarize, these are three types of polysaccharides, long chain of sugar monomers linked together, and these three polysaccharides are all made up of glucose monomers. Okay, but the glucose are kind of linked together in different ways. For starch, it is going to be a single linear branch, linear chain rather, and in some cases the starch will also have some branching, but not too much. And the starch is the storage form of energy in plants. The storage form of energy in animals is going to be glycogen, and we will find glycogen stored in muscle as well as in the liver. And compared to the starch, the glycogen is much more extensively branched. Finally, we have cellulose, which are found only in plants and not animals, and they form these crisscrossing fibers that are super strong, and these are going to be supporting the plants and provide structural integrity to the plants. Here is another example of polysaccharides. There is something called chitin, and chitin is not made up of glucose. It's made up of this kind of modified type of sugar that has nitrogen in it. Okay, so don't worry too much about the chemical structure. I'm not going to ask you to draw them on a test or anything like that. This modified type of monomer is extremely durable and extremely high in strength. They actually form the hard parts of many small animals, such as crab here, the shell, and in some insects where their exoskeleton also contains chitin. We actually managed to figure out how to use chitin in a medical sense. When you say need stitches, the stitches that they use now is actually made up of chitin. It's very strong, but the best part about it is biodegradable, so you don't have to remove the stitches anymore. Once they stitch you up, over time your body will actually break it down, so that helps with the healing process, and it actually will minimize the scar that is left behind from the stitches. The next macromolecule we are going to be looking at is lipids. Just like carbohydrates, lipids consist of carbon, hydrogen, and oxygen primarily. There are some phosphate in certain type of lipids, but for the majority of the lipids, it is going to just contain these three elements. And unlike other macromolecules, lipids are not going to be made up of long chains of monomers, but rather we have these special components that we can put together to form the lipids. And I'll show you some examples of that coming up later. All the lipids are hydrogen. hydrophobic, meaning they're not going to be able to dissolve in water, and so there are some ramification of that. We will discuss that in the slides coming up. In terms of the functions of lipids, you can use lipids as energy storage. Here you can see some fat tissue. We call them adipose tissue. In the adipose tissue, there are these adipocytes, and they are going to store the fat as energy. So if you're running low on glucose, on blood sugar, you are able to metabolize the fat and therefore convert some of them into glucose for use. Lipids are also going to be a key component of cell membrane. We'll be looking at the structure of cell membrane in lecture 4. Lipids is able to form water-resistant barriers. So if you look at an apple, it looks shiny on the surface. That's because of waxy coating, and wax is a type of lipids. So where do we get fats from our food source? Fats are kind of everywhere. You can get fat from most of the food that we consume. Some are higher in fat, other are not so much. Things like butter and oil are definitely very, very high in fat content. And there are actually some good fats. We cannot survive without fat actually, and there are some fat that that are essential for our bodily function. So we call those essential fatty acids. Examples of essential fatty acids are omega-3 and omega-6. These are the ones that we cannot make in the body and must rely on the food source to get them. So you can get high omega-3 from fish, for example, and there are actually some good fats in avocado as well. There are different types of omega-3, EPA and DHA. In fact, these are very important for brain development in child. So the mother, once they know that they're pregnant, doctors would recommend taking some of these as supplements, and even for kids, they recommend having a diet that is rich in these omegas, fatty acids, because they're essential for development. So there are 4 types of lipids, I think, that we're going to look at. The first one is fat. Okay, so fats is an example of lipids, and fats are actually also called triglyceride. Right? So a fat molecule is made up of 2 different components. We have one glycerol, so that's this molecule here. People sometimes call this the ho molecule because of the HO here. So these are the parts of the glycerol that's going to be connecting with the fatty acids. Okay, so a fat molecule is made up of 1 glycerol and 3 fatty acids. So a fatty acid is basically a long chain of hydrocarbons. Don't confuse hydrocarbons with carbohydrates, right? Carbohydrates are the carbs that we talk about with the CHO. A hydrocarbon is just carbon with hydrogen bound to it. So these fatty acids have a long chain of hydrocarbons tail. This one is relatively short. Typically, they're supposed to be 12 carbons or even 16, 18 carbons long. And at the front of it, there is going to be an OH group. So the glycerol is going to connect with the fatty acids, and therefore you're going to remove a water molecule here, remove another one here, and then another one here. So when you form the fat molecule, you will be removing a total of 3 water molecules. And this, of course, is a dehydration reaction, also known as condensation reaction, right? And if you consume fat and you need to break it down in the body, the reverse of that is going to be a hydrolysis reaction. So fat, 1 glycerol, 3 fatty acids. The kind of fatty acids you have in the fat molecule is going to determine whether it is going to be a saturated fat or an unsaturated fat. Let's take a look at saturated fat first. In saturated fat, the fatty acid tail contains carbons that are bound to a maximum number of hydrogen. So let's just go back to this diagram briefly. You can see that the carbon here is bound to 2 hydrogen. Generally speaking, if we have a carbon in the middle of a chain, it's going to be able to bind to a maximum of 2 hydrogen. If you have a carbon that is at the end, a terminal carbon, then it can bind to 3 hydrogen. So if all the carbons in the fatty acids are bound to as much as hydrogen as possible, then we call that a saturated fatty acids, because it's saturated with hydrogen, right? So these saturated fatty acid tail will make the fat a saturated fat. Saturated fats are typically solid at room temperature, so you can think of like a butter. That's an example of saturated fat. Most animal-derived fats are going to be saturated fats. So as you can imagine, saturated fats are actually not too healthy, not healthy actually at all, because they packed really easily inside your body, and they might contribute to clogging of arteries and things like that. So that definitely could increase your risk of cardiovascular diseases. So if you could avoid saturated fats, you probably should. On the other hand, we have unsaturated fat. So unsaturated fat, it's kind of the opposite. The fatty acids in the fat molecule, at least one of them is going to have carbons that are not attached to the maximum number of hydrogen atoms. Okay, there is going to be some double bonds in it. So let me go back to this picture to show you. As I mentioned earlier, the carbon that is in the middle of a chain should bind to a maximum of 2 hydrogen. And in this case, you can see the carbon only binds to 1 hydrogen instead of 2. And the other bond that was supposed to be bound to a hydrogen is now connected to another carbon. So this, in chemistry, we call that a double bond. When you have a double bond in the fatty acid molecules, that will make the fat molecule an unsaturated fat. So if I were to draw saturated fat, it would be the glycerol, and then it would have the 3 straight fatty acid tails. If I am to draw the unsaturated fat, we would have the glycerol, but now we would have 2 straight fatty acid tail, but then the third one would have a kink in it. Okay, so this kink is basically a bend that is created by the presence of the double bond. So what happens is, if you put 2 saturated fat molecules next to each other, they can get close to each other easily. But if I put 2 unsaturated fat molecules together, they won't be able to come together too closely because of this kink, right? It's like trying to ask people to line up one after the other, but then now you tell the person to stick out their arm, right? If everybody sticks out their arm and their feet, then you won't be able to pack as many people together as you would otherwise, right? So for that reason, unsaturated fats, they don't pack together too much, and they tend to be liquid at room temperature. Most plant-derived fats are going to be unsaturated fat. So if you think about canola oil, corn oil, peanut oil rather, these all tend to be liquid at room temperature, and they're considered as unsaturated fat. And compared to saturated fat, this is typically the healthier choice. Here is a side-by-side comparison of saturated fat and unsaturated fat. They both have 1 glycerol molecule and 3 fatty acids. The difference is the kind of fatty acids you have. So if they are all saturated fatty acid, then this is a saturated fat. You can see the carbons are all bound to a maximum number of hydrogen. There are no double bonds in the fatty acids. There are no kinks in the tails. So this is going to be what the fat molecule looks like. And you can pack these very close to each other. In the unsaturated fat, we have 1 or more of the fatty acid tail being an unsaturated fatty acid, and they would have 1 or more double bonds in them, and therefore you can see multiple kinks in the tails, and these don't pack very well, therefore they're liquids at room temperature as opposed to solid at room temperature. I mean, there are some exceptions. If you look at coconut oil, coconut is a plant-based. It's a plant, of course, and coconut oil is a plant-based fat, and those are actually solid at room temperature. but generally speaking, right, unsaturated fats are liquid at room temperature, but saturated fats are solid in room temperature. So there are also trans fats, and trans fats are actually byproducts of a process called hydrogenation. Right? So in hydrogenation, you are trying to add hydrogen to the unsaturated fat to convert them into a saturated fat, and that is how you make things like shortening and margarine. But as a byproduct of this hydrogenation, you will generate trans fat, and we don't have the necessary enzyme, the chemicals to break down trans fat in the body, and so they just kind of keep on building up inside us, and consumption of trans fat has been linked to increased risk of heart diseases and diabetes, and actually trans fat is so bad, but it's used very prevalently in the food industry because they make the food taste better, they increase shelf life of the food. So in a lot of the fast food, in a lot of the processed food, there used to be quite a bit of trans fat, but it's so bad that the Canadian government decided to completely ban trans fat in 2018. So now it's going to be illegal to have trans fat in the food, and even the fast food that you consume will no longer contain trans fat. So that's a good thing. So that's it for fat or triglyceride. That's the first type of lipids. Now we have another type of lipids called the phospholipids. Phospholipids looks like this. There is a head that is going to be a hydrophilic head, a water-loving head, and that's going to be a polar portion of the molecule. And then we have these long fatty acid tail. Okay, so just like the fats that we talk about, these tails can be saturated or unsaturated depending on the type of phospholipid we talked about. So these phospholipid molecules actually is the key component of your cell membrane, and they arrange themselves like so, forming what's called a phospholipid bilayer. So you would have one layer of phospholipid here and another layer of phospholipid here. The polar heads will be either facing the outside of a cell, over here, down here, or the inside of the cell, and sandwiched between them would be these hydrophobic tails, and that will allow the cell to regulate what goes in and what goes out of the cell. And again, we will be talking more about this in later lectures. So phospholipids, it's a little bit special compared to other types of lipids because in addition to CH and O, it also contains P, phosphorus, right? And that is actually the central component of the phosphate group that forms the hydrophilic head. Here is another view of the phospholipid bilayer. We have the hydrophilic heads either facing the outside of the cell or the inside, because those are going to be aqueous environment. They are made up of water, and in between the heads are going to be the hydrophobic tails. So think of this as like a sandwich, right? So you got your bread on the outside and a thick layer of, say, cream cheese in the middle, and that regulates what goes in and out of your cells. The third type of lipids that we're going to look at is wax. Wax is a water-repellent mixture with long chains of fatty acid tails bond to long chains of alcohol or carbon rings. So alcohols is basically molecules with OH in them. We kind of see examples of these already when we talk about how monomers join together. But alcohol is basically a functional group shown here as OH, and there are going to be some carbon rings. So together they make the wax, and wax typically cover exposed surface of plants to prevent water loss through evaporation. That's why if you put a drop of water on a leaf's surface, they don't really soak right through, right? They just kind of form these droplets on top of it. That's because it's coated with wax. It forms what's called the cuticle in plants. Wax also protects and lubricates our skin and hair. So similar to the plant, when you put water on your skin, they don't go through immediately, right? I mean, if you stay in the bath for too long, then you might soak up some of the water. But generally speaking, if you just pour water on yourself, then they don't get absorbed, right? So that waxy coating kind of provides a little bit of water repelling property. And of course, wax is also found in things like in honeycomb. The last type of lipids that we are going to look at would be steroids. Steroids belongs to a group of fats that contains fused carbon rings. So this is cholesterol. Cholesterol is an example of steroids. So each of these rings, the corner of the rings actually represent a carbon molecule. So there's 1, 2, 3, 4, 5, 6 carbons here, fused with another carbon rings. There is an OH group, right? So that's considered as an alcohol. These steroid molecules are going to be important for various biological reasons. First of all, cholesterols are found in your cell membrane as well. They help control the fluidity of your cell membrane. By having a bit of cholesterol in it, it makes sure that the cell membrane is not too solid. But you don't want to have too much. Okay, too much cholesterol will make it too fluid, and it's just going to be too leaky. We also need cholesterol to make different types of steroid hormones in the body. We can use cholesterol as a starting material to make hormones like estrogen in female and testosterone in male. And you can see that there are these fused ring structure again. That's going to be the key thing for you to be able to tell that this is a steroid molecule. We also find cholesterol in things like biosalt, something that we use for digestion of fat, and also cholesterol is important for making vitamin D. So that's it for lipids, and I think that's it for this video. We talk about the relationship between monomers and polymers. We talk about carbohydrates. We talk about lipids. In the next video, we are going to finish the lecture by talking about proteins and nucleic acids. Thanks for watching.